personalized medicine in pancreatic cancer in the era of personalized medicine. This is an unmet need, pancreatic cancer. We'll speak about the overall aim of the project, specific aims, progress to date, data collection, database creation, and the future aims. These are my disclosures. So within this decade, pancreatic cancer is to become the second leading cause of death in the United States. So this is not just a personal problem as a physician scientist treating pancreatic cancer. This is going to become, and already is, a global world health problem. To date, this is the very small arsenal of drugs we have for treating pancreatic cancer with advanced disease. We have made small incremental steps, but not enough. In orange, one can see all the chemotherapies and biological agents, and in green, we can see the biological agents that have been given per specific mutation in the tumor. However, if you think about pancreatic cancer with the patients with advanced disease, and this is the majority of our patients, there are two types of patients. The majority of our patients live for about 6, 7, 8 months, something like that. However, about 50% of our patients, you can see here, 50% of our patients die within 2 months of diagnosis. However, there's also a long-term survival group. So there's a group of patients that are living for more than 12 months or more. So there's a small group of patients that are actually doing better with pancreatic cancer over the course of time. We know that some of the patients in this group that are living for longer and longer have a specific mutation in the DNA repair pathway, specifically germline BRCA mutations. Germline BRCA mutation is seen about in 6 to 7% of patients with advanced pancreatic cancer. And we and others have showed that retrospective and phase 2 data, that when we expose these patients to DNA damaging agents because of their specific BRCA mutations, their tumors are very, very sensitive to platinum treatment, and they can actually have extended overall survival when exposed to specific platinum chemotherapy. However, the toxicity profile of chemotherapy over the course of time is cumulative, and we have looked for additional approaches to treating these patients, and this really leads us to thinking about exposing. This is a case report of a patient with pancreatic cancer. One can see his tumor in the lesion of the pancreas and liver metastases, and he is exposed to platinum-based chemotherapy, and then he's exposed to a PARP inhibitor, which has a selective effect on the tumor and has shown efficacy in BRCA patients in other tumors, for example, breast and ovarian cancer, and now more recently also in pancreatic cancer. So now we know that patients with BRCA-associated pancreatic cancer, really, if we give them platinum-based chemotherapy and then we expose them to PARP inhibition with olaparib, they can have an extended overall survival. And these patients are very distinct and very different to the regular pancreatic cancer patient. However, not all patients with germline BRCA mutations behave in the same way. As you can see here on the slide, we have 2 patients with the same specific mutation receiving exactly the same treatment. The patient on the top part responds, however, the patient on the bottom doesn't. So really trying to dive into patients with germline BRCA mutations, we have divided them into 3 types of patients. So this is a swimmer's plot of overall survival on treatment, and you can see that there's a group of patients that never really respond, not to platinum and not to PARP inhibition. And then there's a larger group, and this is really the bulk of our patients that have acquired resistance. What does that mean? That they respond for about 2.5 years, and then their tumors acquire resistance to this combined strategy of platinum followed by PARP inhibition. And then we have super responders, patients who go into long-term remission on PARP inhibition over extended periods of time. This is almost miraculous when you think about pancreatic cancer. So our overreaching aim of the project is actually focusing on these patients with germline BRCA mutation and trying to build around them the most comprehensive infrastructure we can think of dedicated to discovering and translating effective strategies for pancreatic cancer with germline BRCA mutations. So what we've actually thought of doing is taking as many measurements as we can around the patient, and we're going to soon go into all these different measurements and see how that can improve maybe our understanding. So our specific aims of this project is to create a comprehensive pancreatic cancer database on BRCA patients, to explore patients' clinical response to treatment, to interrogate this with their very, very deep genomic data that we have, and to create a patient's journey timeline with all the clinical data. In parallel, to take all our preclinical database that we have on the patients and to interact it with the clinical data. And finally, to perform even better precision medicine by taking the subpopulation of BRCA germline pancreatic cancer, doing a comprehensive database around the patient, analytics and statistics of this database to identify markers, potential markers for response, to decipher more, in a more accurate way the mechanisms of resistance that we know they're there, and to therefore develop novel therapeutic strategies. So how are we doing this? Let's start with the data lake that we are building. So the data lake has several aspects. It has the clinical data, which relates to all the clinical data that you could think about the patient, things that you think now are relevant, and even things that you don't think are relevant to the patient's journey. All that data is going to be captured and selected. We have omic data, very comprehensive omic data on our patients, including mostly whole genome sequencing, and then RNA-seq, proteomics, and metabolomics. And lastly, we have microbiome, also gut microbiome and tumor, and then all the preclinical data we have on the patient. So this slide just reflects the patient's journey that we are building. You can see that we take a specific patient, and we take all the clinical data we have, when he was admitted, when was our preclinical model made from his tumor. This is for the preclinical aspects. When did he receive treatment in the clinic? When did he do imaging? When we made additional preclinical experiments on the patient, and so forth, and we get all this data and we're putting onto a timeline showing the patient's journey. In regard to the omics, so far we have whole genome sequencing on 25 BRCA patients, and also RNA-seq sequencing, and now we're filling out the proteomics and metabolomics. In regard to microbiome, we're taking stool and saliva, tumor, specific questionnaires to the patients, especially in regard to nutrition and antibiotics, and we're also doing longitudinal sampling of the stool and saliva and tumor microbiome. Additionally, on our patients, we have developed preclinical models, and we have done numerous experiments on these preclinical models. We are taking all this data, all this preclinical data that we have on the same patient, and we're doing digitalization into translational medicine. We're taking all this data, capturing it, and putting it also on the patient's journey. And this is what you can see here. This is the different kind of data. For example, we can make a PDX models, we can make organoids, and then we can have this data to convert in order to be established together with all the clinical data. So to summarize, we regard this specifically as a pilot study. Okay, so we chose pancreatic cancer because that's my field of expertise and my passion from a clinical and a translational research point of view. And we chose BRCA-associated pancreatic cancer because we as a team have had some mileage, and we've been part of efforts in improving the treatment for this specific patient population, including the introduction of olaparib through the FDA to be available for the specific patient population. But this is still not enough. We still feel that even though we have quite a deep understanding of the BRCA, the germline BRCA pancreatic cancer patient, we don't have enough data and we don't have enough deep understanding. We keep going back and looking at the places that we think we know. So in this pilot study, building a comprehensive database around the patient with all the relevant and also what we may think of not relevant clinical endpoints that we have taken, all the raw data from the imaging, from the slides, all the data from the preclinical experience we've done together, putting it in the patient's journey in one data lake for us to analyze and perform artificial intelligence, and hopefully through the study to improve and to really find the next options, the next novel treatment strategies for our patients. We see this as a pilot study. We chose the pancreatic BRCA patients because we're more closely intimate with that kind of specific tumor. And we wanted to start with a small confined cohort as we developed this project. But in the future, we will go out to other subtypes in pancreatic cancer and then hopefully to the whole cohort. And piece by piece try and decipher and understand this disease in a more scientific and to give us more deeper understanding and new options. Thank you.